Hola, muy buenos días a todos. Agradecemos su presencia en esta ponencia y continuando con nuestro itinerario, a continuación tenemos otra ponencia y la doctora Iga nos hará el favor de leer el currículum. Gracias.
Hola, buenas tardes a todos. Bienvenidos a esta última ponencia de la jornada del segundo encuentro de investigación en ciencias de la salud. Es para mí un gran honor, de verdad un gran honor, presentar al doctor Benjamín Ralph. Él actualmente está trabajando como postdoctoral en la Universidad de Montreal. Y una breve reseña del doctor. Bueno, él tiene una licenciatura en inmunología por la Universidad McGill, una maestría en ciencias en microbiología e inmunología por la misma universidad y su doctorado también en microbiología e inmunología por la misma universidad, graduado en el año 2018. Él tiene actualmente en su corta carrera, él se acaba de graduar pues básicamente hace 2 años, 3 años, ya tiene más de 13 artículos de investigación publicados en revistas internacionales y de alto impacto, y muy importantemente, Ben ha sido galardonado de más de 9 becas y apoyos para sus estudios de posgrado y sus estancias postdoctorales. En realidad es para mí un gran honor presentarlo. Ben, thank you very much for joining us and giving us your time. It's a great pleasure for me to have you here, and I will let you talk. Thank you very much.
Thank you very much, Iodine, for inviting me. It's a great honor to have actually been invited by you. Being, my first mentor and introduction into a university lab, it makes me really happy to actually be here today to talk to you guys and present to you my presentation on antibodies treating COVID. While I bring up that presentation. The first half of my presentation is going to focus on what are antibodies and how are they made. I was told you guys are a general diverse audience, so I want to make sure everybody's on the same page for the second half of the presentation, which will talk about how antibodies are used and manipulated in the battle against the COVID pandemic. So I know Irazu presented me pretty well. I didn't quite catch every word she said since my Spanish is not very good. But I'm a McGill graduate. I've studied there for 12.5 years, researching host pathogen interactions, with my PhD work focusing on developing vaccines and antibodies against invasive pulmonary fungal infections. And my current work is I'm doing my industrial postdoctoral fellowship with the University of Montreal and a company called Medimabs. Medimabs is a fee-for-service company that's based in Montreal. It's an antibody-based company, or a company that specializes in antibodies, where we do everything from making an antibody to conjugating it to a fluorophore and everything in between. So this word antibody, I figured that's a good place to start. What are antibodies? Antibodies are proteins that are secreted by a type of cell called the B cell. It's in the immune system. It's a adaptive immune cell, and it consists of 2 chains, heavy chains and light chains. And these molecules are able to bind to target molecules by use of their variable region, and the constant region here provides an identification of their isotype, which contributes to the location of the antibody and the role of it. So a little bit more about the variable region, that is these red regions on the antibody. This is the portion that will bind to a target. Initially, before a B cell is activated, when they haven't seen a target for the first time, these regions are mainly hard-coded in your DNA. They don't have any mutations. They come out as they were initially programmed. After a B cell encounters its target for the first time, the B cell will start to mutate these regions and really increase the affinity for the antibody for its target. At the same time, this activated B cell will take these constant regions and swap them out so that it will change the isotype. I'll get into that a little bit more later, so don't get too hung up on that. But these constant regions are what really provides the information of what function the antibody will have, and where that antibody is going to be located. So here's 3 isotypes that exist, IgG being the most known antibody and the one that people think about when they think about antibodies. They consist of 2 heavy and 2 light chains, and they mainly exist in the serum, existing as this Y-shaped antibody. Then we have IgA antibodies. They exist with 4 heavy, 4 light chains joined together by what's called a J chain. And these are mainly found secreted, so in the intestinal mucosa or in the lung being secreted. And then we have IgM antibodies. These antibodies are in between the two. They can be found in the serum where most of them are, but they can also be secreted. And they consist of 10 heavy and 10 light chains. And as you can see, that's 10 points that they can actually interact with targets even. These IgM antibodies are actually the first antibody that will be produced, and then later on it gets changed to an IgG, an IgA, or well, that's pretty much the 2 that will mainly change to. There's also IgEs, but that's another story we won't get into. How do antibodies work? Well, there are 3 main mechanisms of action. We have neutralization, opsonization, and complement activation. Neutralization is the mechanism where an antibody binds to a target and prevents that target from getting to its destination or causing any problems. So we can think of that with respect to a viral particle. An antibody can bind to the virus and prevent that virus from getting into its host cell. That would be neutralization. Opsonization is where the antibody will bind to the target pathogen and basically flag it for removal from the immune system. It will cause immune cells to come in and actually actively remove it. Complement activation is where an antibody binds to a pathogen and will call in other serum proteins called complement that punch a hole through the pathogen to kill it. And at the same time, complement is able to act a little bit like a flag as well to call in immune cells and help clean up the debris of that dead pathogen. So when are these antibodies actually made in the context of an infection? So antibodies are always going to be present, but in this IgM form, mainly not being secreted by naive B cells, just waiting to see that antigen. So when there's a pathogen that's present, the immune cells will take up the pathogen, process it, and present it to the adaptive immune response, being T cells and B cells. These cells will talk a little bit to activate the B cells further, cause them to start secreting out tons of antibody, initially being these IgMs that are low affinity, not recognizing their target very well. And as the B cell becomes activated, the B cell will affinity mature this variable region to properly recognize its target with high affinity, switching the isotype so that the antibodies will now function as the immune system will need it to function. And these will get secreted en masse into your body, or out of your body if that's going to be an IgA antibody. So now we know what antibodies are, how they help us out when they're inside us. Can we manipulate them for use in a lab? And the answer is, well, as the slide shows, a resounding yes. We use antibodies in a lab to detect the presence of a target. We do this by Western blot, or enzyme immunoassays, or lateral flow devices. These are all assays where we can put a target on or a sample on and see if our target is present. We can also use antibodies to detect if 2 targets are interacting together. And this is done where we can use an antibody to pull out a target and see what comes with it. Does another protein come with it? And we can assess that by the methods like Western blots as well. Another method is doing fluorescent resonance energy transfer, which is where 2 fluorescently tagged antibodies on 2 different targets will transfer electrons or photons from each other to activate another one and see if we get an interaction which will demonstrate 2 targets interacting. That's a very complicated method. So if you want to ask questions at the end, I will freely answer a question on that method. Another method would just be to directly visualize a target using immunofluorescence or immunoelectron microscopy. This is where antibodies are tagged with fluorophores or gold particles, and we can use their respective forms of microscopy to actually directly visualize it, where that target is present on your sample. So we can use them in the lab. How do we use them in the clinic? In the clinic, antibodies can be used for both diagnostic and therapeutic purposes. For diagnostic purposes, it would be using similar techniques to in the lab, using enzyme immunoassays or lateral flow devices. These assays can be modified to look for biomarkers for different pathogen infections or for other biomarkers, such as the pregnancy test for a lateral flow device is one of the most common ones that's actually known. For therapeutic purposes, antibodies can be used to both deplete a target or prevent a target from actually acting in the human body, or can be used as a treatment to also enhance an immune response against a pathogen or against something else. So antibodies are used outside of the human body, and sometimes we want to put them back. How do we manipulate them outside? Like, how do they come to us commercially? Well, they exist in two different forms. You have the monoclonal antibody and the polyclonal antibody. The monoclonal antibody is essentially one antibody that can recognize one piece of one target. It usually does it very well. And this is because a monoclonal antibody comes from a single B cell that's either immortalized or had its antibody gene sequenced and is then recombinantly expressed. Because of this, we can produce as much monoclonal antibody as you desire. The downside of this is it's very expensive. Polyclonal antibodies are the opposite, basically, of a monoclonal antibody. In this form, we have many antibodies that are put together, and as a result, we can recognize multiple epitopes on the same antigen. So multiple different pieces of the antigen on the same antigen. These are produced by bleeding an immunized animal and recovering the antibody from the serum. So as a result, you're limited by the amount of serum you recover. When you run out of polyclonal antibody, you have to start over and get some more. The problem with this is you have to validate lot to lot to lot. In some cases, a little bit of antibody is all you need. Polyclonal is a cheaper method and makes sense. Monoclonal antibodies, that's usually for larger industrial purposes or when you know you're going to be using it for a long time. So I'm going to focus the next little bit on what a monoclonal antibody is and how it's made. So monoclonal antibodies are made by one of two methods mainly. You have the fusion-based method and sequencing-based methods. The fusion-based method is the long-established, most common method to actually make an antibody. This is the method that I actually use a lot at Medimabs. I do this probably 2 times a week about. And what we do is we take the splenocytes from immunized animals, and we're able to go through a process to fuse these B cells to come to monoclonal antibodies. I'll describe this a little bit more in depth in a second. The sequencing-based method is a much newer method. It's very expensive to do. And because it's very expensive, it's usually only large pharmaceutical companies that are actually using sequencing-based methods to develop antibodies. And that's because they're developing them for clinical applications, either for diagnostic or therapeutic purposes. So the fusion-based method, this is done where we immunize an animal. We'll immunize the animal a few times so that we can build up a strong, robust immune response. And after a few immunizations and a little bit of time, we will harvest the spleen from the animal. Then isolate our B cells or splenocytes, and using our splenocyte or B cell population, we fuse them with a fusion partner. This can be like sp2 cells or NSO cells. These are immortalized cell lines that, in the presence of PEG, will form these unstable fused cells that have 2 nuclei. These cells can be just T cells that have fused by accident. They can be the B cells. We don't know what they are, and we don't know if they're going to be productive at secreting antibodies. So because of that, we have to isolate out each cell to identify which ones are producing antibodies and which ones are producing antibodies against the target that we're interested in. To do this, there are 2 methods. We have the semi-solid media isolation method and the limit dilution method. The semi-solid media isolation is, as it sounds, it's like a petri dish. You fill it with a media, not exactly agar, but kind of got a little bit of gelatin substance to it going on. And the individual cells will grow to form little colonies on the petri dish. We can then take out these colonies and assay to see which ones are secreting antibodies and which ones are secreting antibodies against the target we're interested in. The limit dilution method is technically less demanding, being that you just heavily dilute your cells and plate them in a lot of 96 well plates, so that each well in the 96 well plate will have only one cell, and there will be a lot of empty cells in this. So there's a lot of waste in this method, but it's technically less demanding. Once we have isolated colonies from whichever method is used, we then screen these colonies, and we screen up to 1,000 colonies at once to look for cells that are secreting antibodies against the target of interest. Once we find some, we have to stabilize these clones. Stabilizing means a lot of these cells will die during the process. They will stop producing antibodies during the process. And the reason why is these cells are not designed to have two nuclei. They're not wanting to exist in this format that we're forcing them to exist in. Because of that, some of them die, some of them stop producing antibody. So after a stabilization process where we allow them time to get accustomed to being these fused hybridoma cells, we will finally freeze the good cells. And at the end of all of this, we hope that we're going to have at least 10 clones from a project. And that's actually a pretty decent number. So we go usually from 1,000 colonies screened to 10 clones from all this work. And that's why I mean this is usually an expensive process. But when you find a good antibody, you're finding one that can possibly be used for many purposes and be actually worth a lot of money. So there is the benefit, but it comes with a strong risk. The other method, there's not much to say at the moment, but it's the sequencing-based methods. There's no gold standard for how anybody does this at the moment because of how new it is. And it's actually a method that the company I'm working at is trying to develop. And that's the whole goal of my project, is to try to develop a sequencing-based method for the company so that the company can eventually offer an affordable option. The goal of this is usually to reduce the risk, sometimes at the expense of extra cost. So now that we know what antibodies are, what monoclonal antibodies are, and what that means, how are antibodies being used in the battle against COVID? So I don't think I need to introduce COVID too much unless people have been living in a cave somewhere. But there's this virus called SARS-CoV-2. It's been causing a pandemic around the world for the past 2 years about, and has been the reason for about 4.6 million deaths in that time. The important thing for this presentation is that there are many variants that have come about during this period of time. And that's because every time the virus mutates, antibodies may not recognize what they were designed to recognize because of those mutations. So how I think of COVID when it comes to treating the infection is the antibodies are almost running a hurdles race. They're trying to overcome all these variants that are coming up, where the antibody is trying to overcome another variant that's trying to get into its way from actually being an effective antibody to battle COVID. So as I said, the virus would be kind of like the hurdle, and our contenders in this would be the 3 dominant treatments. I decided to go with the 3 dominant treatments in the United States and with their information, just because I'm in Canada, presenting in Mexico, and between us is the States, so it seems appropriate. So the 3 main treatments are Regencov, Cevudi, and Blimey Nevimab. Regencov is a treatment that's made up of casirivimab and imdevimab. These 2 antibodies are both monoclonal antibodies. So 2 monoclonal antibodies were put together for the establishment of this treatment. They're developed by Regeneron Pharmaceuticals, and they recognize the spike protein on the virus. actually all the treatments I'm presenting will recognize the spike protein. That's because that's the protein that is used to get into your cells, and by targeting the spike protein, you're hopefully preventing the virus from getting into your cells. Remember the method called neutralization that I presented? That is the method that is hypothesized these antibodies are actually acting under. And by using two antibodies, it was thought for this Regencov is that it's going to help prevent it from not being made effective at future variants. Our second contender, Cevudi, is also known as sotrovimab, was developed by GlaxoSmithKline and Vir Biotechnology. Again, recognizes spike protein, but this is our oldest candidate in the batch. This one was actually developed against SARS-CoV-1, the early infection in 2003, and is actually still effective against SARS-CoV-2. So it's, even though it's the oldest antibody, it's still pretty good contender in this hurdles race. And then our last one is Blimey Nevimab, which is just like Regencov in that there's two antibodies that's used in this treatment, the other one being etesevimab. So again, two monoclonal antibodies that are mixed together for this treatment. The Blimey Nevimab is made by AbCellera Biologics and Eli Lilly. The shout out to AbCellera in this, while being the competitor to Medimab, the company I'm working for, is that AbCellera is also a Canadian company, so that's the one shout out there. And Junshi Biosciences makes the other component, etesevimab. And again, all these antibodies recognize the spike protein. So how effective are these antibodies? Well, in their respective trials, they were all found to reduce hospitalizations when they were given to patients by 70% and up. So they're all actually very effective antibodies for preventing hospitalization in patients. So let's go through the variants now. As I said, these variants are presenting problems to these antibodies and being effective treatments against the infection. So the alpha variant came along, and all these antibodies were actually found in vitro and in patients to still be very effective as treatments. When the beta variant comes along, Blimey Nevimab was found to have a marked reduction in vitro, and eventually this was true in patients as well. For the gamma variant, the same remained. Blimey Nevimab was still showing a significant reduction in the in vitro assays, and the same was coming true in patients. Now we get to the Delta variant, the one that became the dominant variant to infect patients, at least in Canada and the US. This variant showed only a modest reduction in vitro at the beginning stages and was unsure how it was going to be as a treatment, but later data found that it actually wasn't that bad and is actually a pretty decent treatment still. All the other antibodies still to date, Regencov, Cevudi, are acting pretty well. Then we have the epsilon variant that demonstrated a modest reduction and still demonstrates a modest reduction, and the same for the iota variant. Now, these are the variants to date that good data exists on. We also have the kappa, lambda, and mu, and at the time that I made this presentation, there wasn't any really good data for me to refer to actually say how these antibodies would work with in response to these variants. Maybe that data has actually come out and I haven't seen it, but there's going to be more variants to come, and these antibodies are not done with their hurdles yet. So in a little bit of a summary for the treatment using these antibodies, Regencov and Cevudi have basically come out on top with being effective treatments against every variant that they've come across. Blimey Nevimab was strong against the alpha variant, but the beta and the gamma were hurdles for that. They had a hard time with those ones. And for that reason, actually, in the States, it stopped being recommended as a treatment for a while and was stopped being distributed. Then for the Delta variant, it started distribution again, and it's still actually now being used as a treatment. So treatments are great, obviously. But what else is there that the antibodies can be used for? Or, sorry, I'm getting ahead of myself. Besides the monoclonal antibodies as treatments, we have polyclonal antibodies. Remember, polyclonal antibodies come from the serum of an immunized animal. In the case of treating patients, we can't use serum from other animals. It has to come from humans. So it comes from usually previously infected patients. The problem with using plasma to treat a patient is that there's a lot of possible adverse side effects. We have the possible transfer of infectious diseases like HIV and hepatitis B. You have the possible proteins inside the plasma that can cause febrile non-hemolytic reactions or hemolytic reactions. So all these adverse side effects, it has to be balanced against the possible benefit that you're going to gain from transferring plasma from one patient to another. And because of that, like the NIH guidelines were not clear on whether the benefits outweigh the risk of the adverse side effects. So really, polyclonal antibodies for this might not be a strong solution, except for the fact that by technicality, Regencov and Blimey Nevimab are actually polyclonal antibodies in that there's two monoclonals mixed together, but let's not get caught up on technicalities. So beyond the treatment, as I was saying, you have treatment, you also have diagnostics. And monoclonal antibodies are employed as diagnostics against COVID. You have the rapid antigen detection kits that exist. So these are lateral flow assays that you put a sample on and you're able to find out whether the sample contains viral particles or not. Because of this, it is a good assay to determine whether a patient is infectious or, well, they're not very sensitive, so it doesn't rule out that they're not. So they are less sensitive than PCR, but they're much faster than the PCR test. They take 15, 30 minutes to employ versus hours for a PCR test. So how does a lateral flow device actually work? Well, you put a little sample on that contains what you're trying to figure out if it has the virus, and you let it flow across a membrane. As it flows across, your sample that was entered here or here will eventually come in contact with a lot of antibody that has a tag on it that we'll be able to visualize later on. The sample will continue flowing across and will encounter immobilized antibodies that will also recognize your target of interest. So in the case of the virus, you have viral particles that will now be tagged with antibodies that have a tag on them, and they will get held back at this line, being the T line or the test line. And then the free antibodies will continue, and they will get held back now at the control line as a control for the test to make sure that your sample flowed across the membrane and you have a control line to visualize that your tests happened correctly. If you have the virus present, you would get a line at the test line. Now, this test has a blue line, which strongly suggests that they're using the latex bead method coupled to their antibodies, but you have other things you can couple, like gold or fluorophores. Fluorophores are not so common, gold particles are, and they will usually give a reddish tinge to your assay. So, in summary, and I think I went a little bit too fast. I am sorry because I talk very fast and I didn't mean to go through my presentation so quickly. But in summary, antibodies or monoclonal antibodies make an effective treatment against the COVID infection. But we have to be careful when mixing the right clone with the variant that's infected, because that will dictate whether the treatment will be effective or not. And then they also, monoclonal antibodies actually make a pretty decent treatment, pretty decent diagnostic tool as well in the lateral flow assays. So in closing, I want to thank Medimabs for giving me the opportunity to work at that company and do the research I do. The professor I work with at the university is Dr. Brian Willem, and I am thankful to be able to work on his team as well. And my tax is the funding that makes this all possible for me. And then, of course, I would like to thank you guys for inviting me to give my presentation today. Are there any questions? I will stop sharing.
Hello, Doctor. Thank you for your presentations. It was a very interesting presentation, and we are seeing if there is some questions, but well, I think we don't have questions. So Well, we don't have any questions, so we are going to read the certificate. Well, this certificate that Benjamin Roth participated as a guest speaker in the second international meeting of research in health science with the conference entitled Antibodies Treating COVID in November 12, 2021, Patria, Ciencia y Trabajo. And Doctor, thank you for your conference. It was very interesting.
Thank you very much.
Thank you. Y a todos los participantes les queremos dar las gracias por seguir aquí. Les vamos a mandar el código por el chat, porque recuerden que tienen que guardarlo. Y pues eso sería todo de nuestra parte. Y a nombre del comité organizador, les queremos dar las gracias. Y esperen un momento. Vamos a esperar aquí un poquito. No sé si la doctora Iga esté por ahí, pero... Ok. Doctor, dicen que there are some questions.
Sure.
We are going to wait until they arrive. Bueno, mientras a los participantes, les vamos a ir anotando en el chat el código que tienen que guardar en lo que nuestros compañeros escriben las preguntas.
Well, thank you very much, Ben, for your presentation. It was really very new things for some of us. There is a question. I don't know if you can see it in your screen.
I can see one in the private chat. I think antibody treatment will eventually be used as standard therapy for COVID patients. My hope is yes, but I don't know. I would like to see it because of how effective it has been in a lot of the trials that I've seen and in what the data shows, but I don't know if it'll ever be a standard treatment. I guess time will tell on that. It's also a very expensive treatment because producing these antibodies is actually very expensive. Oh. To start investigations. I'm not sure what you mean by which topics do you recommend to start investigations. Could you elaborate?
Ren, ¿podrías, por favor, replantearle la pregunta al doctor? O si la quieres hacer en el chat para que el doctor Estrada o yo podamos hacer la traducción.
Oh, it's difficult to say. Yeah, that is difficult. I would say if you have something you like, a topic you like, and you want to do research in, then maybe the best way is to find somebody that's doing that research in the field and to contact them in advance to find out if they have space on their team. I mean, often a lot of labs are always filled to the brim with as many as they already can take. But it's usually a matter of luck of being the right person to ask at the right time. Oh. Then I'm sorry, what do you mean?
I think it's about the last question, that which topics do you recommend to start investigation or research as a student?
What do you mean by which topics?
Or how...
Just give me 1 minute, please. Les voy a pedir a los chicos que tengan preguntas que las puedan mandar al chat y no las manden, por favor, a nosotros para que las podamos traducir y se las podamos dar al doctor. En el medio tiempo, I have a question, Ben.
Okay.
A few weeks ago, when we talked about your work that you were doing in your company, one of the things that I remember is that you told me that your company makes personalized antibodies. So one person comes and tells you, I want this antibody. When you prepare the antibody and you give them to them, which are the applications for those antibodies?
Oh, yeah. Really, a lot of clients ask for them for almost every purpose you can imagine. We've developed antibodies for the livestock industry, where I can't say exactly what they're doing with them, but there are antibodies actually used in the livestock industry I didn't imagine. And then a lot of times it's for a lot of labs where they just are missing a tool to continue doing their research. So we've made a lot of antibodies for obscure targets that very few labs work on, but they need a tool to be able to do their work. And then some companies have come to us to develop antibodies to see if they were going to find one that will be used in a treatment or a diagnostic tool. They will come to us and we'll give them the tool, and then either the company sometimes shelves it and uses it internally, or they'll end up selling it.
And I know that you cannot tell us many details because you work in a private company, but around how much is a monoclonal antibody? If I go with you and I tell you I need this.
Animal work is about 3,000 Canadian, and the fusion part is 2,500 to 3,000 Canadian as well. So it's like a $6,000, $6,500 job, usually. Yeah, that's against the protein target. And that's where we have to do all the synthesis ourselves as well, and finding the right parts of the target to target. Cost could go down by up to $1,000 maybe if the targets were provided to us and all we had to do was the immunizations on our end and then the fusions.
And this new technology that you talk about in your presentation about the sequencing-based method, your company is able to do that method, or is?
Hopefully in 6 months, I can say. That's the project I'm working on, and I'm finalizing hopefully the method, and hopefully in 6 months we'll be able to properly market that method.
And it's going to be a better method to generate monoclonal antibodies?
Yeah. The goal of that is to minimize risk, because doing the standard method, it's, as I said, like $6,500 Canadian, and at the end you might have nothing, which is a lot of money that will be wasted. But a sequencing-based method would hopefully minimize the risk, because you sequence a pool of cells and you're trying to use a lot of this data to find if you can pull out the antibody just from that, the correct sequence that's going to recognize the target.
Okay, thank you. Just let me see if some of the audience have another question before we say goodbye. There is another question there. Sure. I think that you can read it.
Yeah, can I? Oh.
Please read the question, because the audience cannot see the chat.
Yes. Okay, that's true. Have there been any reports on whether synthetic antibodies are more specific or have higher neutralizing capacity for COVID proteins than natural antibodies for patients in vivo? Ooh, that's a good question. I don't know if there are any reports on that. Well, I can't say if there are any reports on if synthetic antibodies are more specific for COVID or more effective. The ones I know of, the ones I presented, they are all natural in a way because they were identified from patients previously, and they were identified using sequencing-based methods, actually. But other than that, there is no modifications done to those antibodies to really qualify them as being synthetic. So I don't know if there's any heavily modified or synthetic or fully synthetic antibodies yet that will be effective. Sorry, can't give you better information than that.
Ben, and I have one last question. I would like to have your insight, you as immunologist.
Yeah.
What is the future of monoclonal antibodies for treating many different diseases? So now we have them for treating some, for example, cancer or some other diseases.
Yeah.
And they are not fully used, like they are used like a coadjuvants of some other treatments. But you, as an immunologist, what do you think that is going to happen with monoclonal antibodies in the future?
Well, I think because of personalized medicine and a lot more obscure targets, particularly with diseases like cancer, where it varies so much from patient to patient and different biomarkers that have to be targeted, I think there's going to be a lot more antibody-based therapies to target complex diseases like cancer, where you can have one marker that's upregulated in one patient but not upregulated in another because it's another set in another patient. And there are many ways that we can modify antibodies such that we can target two targets simultaneously on the same cell, or we can target one cell and using a modified antibody, we can pull another cell in proximity to actually bring them close so that hopefully the immune cell will kill that cancer cell. I'm using cancer as the example, but there are many diseases where this can take place as well. So I think antibodies are really up and coming or will be utilized more in the future.
Thank you.
Thanks.
Ok, ahora sí. Entonces, si ya no hay más preguntas. Well, Ben, thank you very much. There is no more questions. And it was a real pleasure to have you here. And thank you very much for joining us. And I will let you, I will send you your certificate of participation in the next few days because the one that we showed you is not the final. And thank you very much again. I will send you an email very soon. Thank you.
Thank you very much for inviting me. It was a pleasure presenting today.
Ok, chicos, pues ya si quieren podemos pasar a la parte de la premiación de los pósters. Ben, you can disconnect if you want.
I wasn't sure. Okay.
Thank you.
Okay. Bye.
Bye. See you soon. Ok, tenemos ya a los participantes de los pósters.
Pues creo que no podemos saber porque no salen los que lo están viendo, pero no, o sea, en el stream ellos no están. No sé si gusta que les mandemos la liga y nos esperamos un poquito.
Nos esperamos un poquito. Renata ya les está mandando mensaje de que ya va a comenzar la premiación.
Vale.
Ok, buenas tardes a todos otra vez. Pues bueno, llegó el momento de finalizar nuestro segundo encuentro de investigación en ciencias de la salud y vamos a terminar nuestro evento anunciando los ganadores del concurso de carteles que se presentó a las 12 del día. Tuvimos 7 carteles participantes en la categoría de pregrado, solo de pregrado, y no sé si ahora sí me puedan apoyar los chicos de OEPSA para proyectar la constancia del tercer lugar, por favor, para que lo podamos anunciar. Bueno, les comento que la evaluación fue hecha por un trío de jurados, en donde se hicieron bajo una rúbrica y se hicieron los promedios de dichas rúbricas. Y entonces nuestro tercer lugar fue para el cartel de investigación original que lleva por título Análisis del acoplamiento cardiorrespiratorio en mujeres con y sin diagnóstico de preeclampsia, presentado por Elías Pichardo Carmona. Entonces, no sé si por ahí esté Elías Pichardo Carmona. No, bueno, entonces vamos a hacer el anuncio del segundo lugar. Por favor, me pueden pasar la siguiente diapositiva del segundo lugar, que fue para el cartel titulado COVID-19 en niños, y lo presentó Leslie Moisen Vilchis. Y por último, el cartel ganador, si me pasan la diapositiva, por favor, fue para el cartel que llevó por título Guillain-Barré, variante Miller-Fisher posterior a inmunización contra SARS-CoV-2. Este fue una revisión de caso y lo presentó Dulisa Reyes Salinas. Entonces, pues, muchas felicidades a los 3 ganadores. Se les va a hacer llegar su constancia por la organización de OEPSA. Los que no resultaron en los 3 primeros lugares, de todas maneras, también les externamos nuestra felicitación y que ojalá y sigan participando en este tipo de eventos científicos que son tan importantes para la formación de nuestros estudiantes en las áreas de las ciencias de la salud. Entonces, si no tienen ningún otro comentario o alguien quiere, de los chicos de OEPSA que quiera tomar la palabra para hacer la clausura formal de nuestro evento.
Sí, doctora.
Adelante.
Sí, bueno, queremos agradecer a todos los participantes y muchas felicidades a los ganadores de los carteles y a los que no ganaron, pues también muchas felicidades, porque como dice la doctora, es muy importante para nuestra formación y a nombre de todo el comité organizador damos por terminado toda nuestra jornada de actividades y les queremos dar las gracias también a usted, doctora. Muchas gracias. ¿Sí me escuchó?
Sí. Sí, gracias. Nada más, bueno, ya para hacer la clausura formal, bueno, a nombre de lo que es el Cuerpo Académico de Neurociencias, el comité organizador de este evento, el doctor José Antonio Estrada Guadarrama, el doctor Luis Ángel Zapicolín y una servidora, con colaboración obviamente de la Organización Estudiantil en pro de la salud de la Universidad Autónoma del Estado de México. Les agradecemos la presencia en este evento y siendo el 12 de noviembre del año 2021, siendo las 17:59 horas, damos por terminadas la jornada científica y académica del segundo encuentro internacional de investigación en ciencias de la salud y esperamos que el próximo año podamos hacer el tercer encuentro. Muchas gracias a todos.
Muchas gracias, doctora.